We're entering the new era of individual genome sequences. I put this as a separate category from the Human Genome Process project, and I'll show you why in a moment. And then disease gene identification. So let me just say a word about each of these 5 things briefly. So first of all, the Genome Project gave us a reference sequence. We called it quits at 2003, said we had the reference sequence. And then we started saying, well, how does each person differ from the reference? And we now know a lot of different ways that each of us differ genetically from the reference sequence. The bottom line is each of us has about 3 million variants from the reference sequence, a different set of 3 million variants from the reference sequence. And we have a lot of these things that are called copy number variation, which you'll hear about from Hamish this afternoon. So a lot of variation one person to the next. That's no surprise. We can recognize everybody as being different in our species, so we should have expected that at the beginning. There's a big project going on now, the so-called 1,000 Genomes Project, and many of you know about that, but that's something to pay attention to. There'll be a genome biology meeting next week, and the 1,000 Genomes Project will be meeting on Monday and sort of assessing where they stand. But the goal of that is to do genome sequence on 2,500 individuals from 50 different populations around the world. That will end up giving us a good accounting of the common variation in our species throughout the populations of the world. Now, whole genome sequence biology, Steve has alluded to this, but this is really an amazing diagram if you look at it. This was published in April 2010 in Nature. And just pay attention to this red curve right here. So this is the cost per million base pairs of high-quality sequence. So in 2000, it cost about $10,000. Now let's fast forward to 2005, it cost $1,000. Let's fast forward to 2008, it cost $10. Let's fast forward to right now, $1. Amazing. This is dropping at a faster rate than Moore's Law is going up for computer science. So as Steve said, we're under $10,000 a whole genome right now. We can get one on whoever you want for $7,000 right now, and if you have friends, you can get one for $3,000. Compare that might sound like a lot of money, but compare it. How many of you have been to Johns Hopkins Hospital and spent a day in the outpatient clinic? $3,000. If you can walk out under $3,000, that's not bad. So it's in the context of what modern medicine costs, that's not very much. And this information lasts you for a lifetime. So it's a real revolution. This accompanying paper, let's see here, estimating how many whole genome sequences have been done. So at least 2,700 are thought to be completed by the end of this article was published in November, and by the end of 2011, the estimate is about 30,000 whole genome sequences done. So it's a huge change in human biology and medicine, having this information. Now, what about evolutionary thinking in medicine? So, evolutionary thinking, really, I think of these factors. First of all, evolution tells us the centrality of variation. If we think evolutionarily, we think variation, one individual to next within a species. And we understand that natural selection is going on all the time. Think of the calories we all ingested in those good-tasting donuts outside. Evolvability in systems biology. In other words, how do biological systems respond to the pressure of natural selection? Emphasis on an integrated biology. We've been through the heyday of reductionist biology, and now we realize that we have to put all the pieces back together again and understand how the organism works. That's integrated biology. And individuality. Why is evolutionary biology emphasize individuality? Because selection acts on individuals. So it determines who gets sick and who doesn't get sick, who comes to see their doctor and who doesn't. Now there are many examples, and I don't have time to go into them, but just to mention a couple that I think are really intriguing. One is there are now methods, and I don't have time to go into it, but taking account of the SNP variation of the genome, you can find regions of the genome of Homo sapiens that are undergoing tremendous positive selection right now. So that means that a particular population in a particular part of the world, a particular region and the genes in that region are undergoing positive selection. That means there's something in that region of the genome, a variant in that region of the genome that confers advantage in ongoing natural selection. So those same regions are likely to be, when the person doesn't have the advantageous form, are likely to be responsible to disease. So current estimates put about 300 areas in the genome where there's intense positive selection going on right now. Here's another one. We can understand our relationship to our closest relatives. So you know that the Neanderthal genome was sequenced recently. In this group by Santi Pavo, one of the things that they did is they asked a simple question: what regions of the genome have changed most rapidly between Neanderthal and Homo sapiens? And if you look, you see genes that are in the category of energy metabolism, differences in energy metabolism, a lot of genes that are expressed in the brain and are thought to play a role in cognition, neurodevelopment, and several microRNAs. These are those molecules that regulate gene expression. So that suggests to me, if we want to understand what makes us different from Neanderthal and what are the characteristics of our species, we look in these regions as a good place to start. Now, what about, let's turn now to the individual genome sequences. So this is really, we've been talking about the reference genome. Now we're going to talk about this genome sequence of me or you or our patients. So really the first one that got published was not surprisingly Craig Venter, because he used his genome for most of the Celera effort. So his was pretty much sequenced at that time. And just as I said, in his case there were 4.1 million variants versus the reference sequence, and copy number variants and the variations and so forth. But to put it in a different form, think of Craig. I'm sure you all have seen him. You can know his whether or not he's lactose intolerant. You can know what his genotype is of the DOPA D4, dopamine receptor locus. DOPA D4 is the risk-taking variant, and I haven't looked, but I'm guessing he's got it. And you can also, if you want, look at his ApoE genotype. Again, I haven't done it, but you can to see if he's at greatly increased risk for Alzheimer's disease. So that's a whole different way of looking at patients, asking what are their individual risks. And as Steve said, the illumination right now is pretty dim, right? So we've gone from darkness to dim illumination, and every day the rheostat gets cranked up a little bit higher. Now, what about disease genome? And the way you can see the rheostat being cranked is here. We now know in online Mendelian inheritance, which is run by Ada Hamish, you'll hear from her later, we now know of about 2,500 genes that there are variations that cause disease. That sounds like a lot, but it's only about 11 or 12% of our total complement. So we have a long way to go to find the rest of them. A big change is the number of genetic tests. When I was practicing, well, I'm still practicing, but just a few years ago, there may be 5 or 6 genetic tests. So now, there are 1,700, you can't read this very well, but there are 1,700 genetic tests. So each of those genetic tests gives you a precise diagnosis and gives you insight into differences between one patient and another patient with the same diagnosis. And this is a big challenge for pathology, where all tests are done. So that's, if I was you, you might look into that story. Molecular cytogenetics, you're going to hear from that from Ada. And progress in identifying genes contributing risk for complex traits, you'll hear from that from Fred about diabetes, and Aravinda about sudden cardiac death and so forth. So that's going on dramatically.